Hello, everybody, and welcome back to BioSC 140 Human Physiology. This is the video for Laboratory 13 and 14, which is over EMG and biofeedback. All right, so there are two experiments in this one lab. One is on EMG, which is electromyogram, and one is on biofeedback. So let's get right into it. So for both of these experiments, both of these two labs within one lab, if we were in a live class, we would use something called a Biopack. A Biopack is a laptop computer with like a side processor that we're able to add or connect different types of sensors to this side processor. Heart rate monitors, all sorts of different types of tools to this side processing processor. And we're going to do, I think maybe 4, maybe 5 experiments throughout the semester with this Biopack. We're online though, so we're not going to get too deep into how exactly to hook up all these different wires, but we'll go over what these sensors do and what they record. So we'll skip over those. Perfect. All right, so first off, let's do a little background information. What is an EMG? It's an electromyogram. So let's break the word down. Electricity, myo, muscle, gram is like a graph. So an electrical muscle recording. Know both terms. I'll use both terms, EMG and electromyogram. And what does it do? It measures electrical activity. So the sensor that we hook up to this Biopack processor for this experiment it's a wire with a few like circular pads on the end. And we're going to put those pads on top of our skin. And the pads are going to measure all electrical activity from nerves and muscles underneath those pads. So they're going to measure all electrical activity underneath the pads that we place on our arm. Now, this is a question that students miss pretty frequently, so I'm going to point this out right here again. It's all electrical activity. It's not just nerves. It's not just muscle cells, muscle fibers. It's all electrical activity, and both nerves and muscles will have electrical activity during a muscle contraction. All right. One other thing I want to point out. It does not directly measure strength. It measures electrical activity. Now, electrical activity and strength are related. Electrical activity leads to force production, but it is not the exact same thing. So please keep those separate in your minds. So right here in this image, you can see what the setup looks like. We have our computer, we have our graph, and we have these sensors on our subject forearm. So we're going to set up the pads like this on some students' forearm. And then we're going to have our student squeeze their fist, clench their fist 5 times. The first time, it's going to be with 25% effort. And we're going to see what the electrical activity underneath in their forearm looks like during 25% effort. Were they going to do it for 50% effort? Were they going to do it for 75% effort? And then we're going to do it for 100% maximum effort. 4 grips. We're then going to repeat the maximum effort grip with a machine called a dynamometer. And what this machine is going to do is it's going to measure the amount of force for their grip strength. How much force do they generate when squeezing their fists together? And then we're going to repeat for the non-dominant arm. So we're going to do all 5 for dominant and non-dominant. What do you think is going to happen to the amount of electrical activity as the subject squeezes harder and harder and harder? How is electrical activity going to change between these different experiments or these different trials? How is it going to change between dominant and non-dominant arm? How is force production going to change between dominant and non-dominant arm? Hypothesize, because we're going to talk about it in discussion. All right, so here it is. We'll see, this is kind of what the data looks like. Different ways of showing the same data. You can see that it's kind of squiggly lines here and kind of a, yeah, so that's what the data will look like. But we'll get into that with discussion. Here are the different trials. Perfect. All right, so that's how we do the EMG electromyogram experiment. Now, let's look into how we do the biofeedback experiment. So with the biofeedback experiment, it's going to be a little bit different. We're going to hook these sensors up right here to a couple students' fingers. So these sensors, these wires are going to go to our Biopack, which is connected to our computer. And what these sensors are going to sense, going to detect, is heart rate. So the heart rate of our subject, and then GSR, or galvanic skin response. Galvanic skin response. What this is it's how easily does electricity travel over a person's skin? How easily does electricity travel over a person's skin? Once we have these set up, we're going to record these rates during 3 conditions, during 3 situations. So our subject, our student, is hooked up to these sensors, and we're going to have them relax. Sometimes students will put on like a peaceful video on a laptop, like a beach with, you know, waves rolling in. We're going to have the person relax, and we're going to see what their GSR and their heart rate looks like. We're going to have them open their eyes and relax for about 30 seconds. And then we're going to have them relax with their eyes closed, max relax for 1 minute to 2 minutes. And then we're going to startle them or get them into an aroused state, a state where basically what we're going to do is we're going to have students try to scare them. The student's going to relax with their eyes open. The student's going to relax with their eyes closed. And then their group mates are going to scream, yell, stomp their feet. Dropping books on the table works really well too. They're going to do something to startle the person. And we're going to see how heart rate and GSR changes when they're startled. We're then going to analyze the data for heart rate and GSR. So EDA down here, the different resources we have, they kind of use GSR and EDA interchangeably. GSR is galvanic skin response. EDA is electrodermal activity. They're just measures of how easily electricity moves across the surface of your skin. It's a good thing to think about. What might affect the ability of electricity to move across the surface of a person's skin? And we're going to record, control, relaxation. So eyes open, relaxed, eyes closed, relaxed. And then as the group mates scare the person by dropping books on the table, stomping their feet, screaming. It's actually pretty funny in class because, you know, people are working hard, they're concentrating, they're relaxing. We usually have 6 groups in the class doing this experiment. And then all of a sudden, you'll have 4 people in a group all stomp their feet and drop books and scream at the exact same time. And it kind of scares every other group, especially the first time it's done. It's a fun lab. So how do you think heart rate's going to change during each of these states? How do you think EDA or GSR is going to change during each of these states, and why? All right, that is the experimental procedure video. Next up, we'll be back with the discussion video for Lab 13 and 14, EMG Biofeedback. Please email me if you have questions.